So today we will be reviewing the key terms used so far, monohybrid crosses, and an introduction into test crosses that will be discussed in the coming week. So these are the different terms that we've used so far. We've got allele, which is a different version of a gene. We've got trait, which is each variable character. Phenotype, so the characteristic of an organism that can be seen, so blue eyes, brown hair. And then the genotype, which is the genetic makeup of an organism and its combination of alleles. We've then got recessive, so that's the allele that's expressed in a homozygote recessive and is carried by a heterozygous, but masked by the dominant allele. We then have dominant, which is an allele that will be expressed in both the heterozygote and the homozygote dominant. So as you can see here, it would be the big B, and in here, the big B, big B. Whereas this one is only present when it's the little b, little b. Okay. We've then got homozygous, which is two identical alleles, and heterozygous, two different alleles. So if you're not sure, this bit here really differentiates. So homo, same, hetero, different. So we're just going to go through autosomal and sex chromosomes. So autosome is a chromosome that is not directly involved in determining the sex of an organism. So we've got 22 here. And then we've got the sex chromosomes, which is a chromosome that determines whether an organism is male or female. So as you can see here, we have a male. If it was XX, it would be a female. So we're just going to go through these terms again using these two homologous chromosomes. So as you would remember from your last section in mitosis and meiosis, you get a maternal, which we'll say is this one, and you get a paternal, which is from your father, so from your mother and from your father. So these have the same genes at the same locus, so the same location on the chromosome, but they might have different alleles. So we're going to use eye color as an example of the different sorts of alleles that you can have. So if you're homozygous dominant, both your maternal and your paternal have big B. So therefore the eye color that is expressed is blue eyes, and they're homozygous because both chromosomes have that big B. If you're homozygous recessive, then you have the two little b's, and green eyes is expressed because that's the recessive trait for your eye color. Finally, you have heterozygous, where you have one big B and one little b. But since the big B is dominant, blue eyes is expressed, and the individual is a carrier for green eyes. So when we're applying this, we use a Punnett square. So that's a diagram used to predict an outcome of a breeding experiment. So you put your gametes here, and then you put your males here and your females here. So we're going to go through a worked example so that you can see the process. So a monohybrid cross is the simplest kind of Punnett square, and it's mating individuals with different alleles but for one gene. So we do this as a way of predicting the offspring without actually having to breed the offspring, and that way we can work out the genetic and the genotype ratios. So males and females only give half of their alleles, so we need to split them up. So your female would give one gamete with a big R and one gamete with a big R. The male would give one sperm with a little r and one sperm with a little r. This example, both are homozygous. So then we move them into the Punnett square. So you've got your big R, and your little r, and your little r. We then add the little r's down, and we put the big R's across. So always make sure you pop your dominant allele in front of your recessive allele. So we started off with two homozygotes, and we've ended up with 100% of the offspring being heterozygous. So they will be carriers of the little r and express the big R. So we're just going to work through this worded example. So if a homozygous blue-eyed man and a heterozygous blue-eyed woman, were to have a child, what are the possible genotypic and phenotypic ratios? So genotypic means the alleles, so whether it's big R, little r, how many big R's, how many little r's, and the phenotypic is how many have blue eyes and how many might have a different coloured eye. So we've been given a really good explanation. So homozygous blue-eyed man and a heterozygous blue-eyed female. So that means that blue-eyed is dominant. So homozygous, same for the male, and heterozygous, different for the female. So we've got big B for the male, and big B, little b for the female. So we separate them into gametes, and we put it into the Punnett square. So we'll move them in now. And then we do the same as last time. So move them down and move them across. So as you can see, we've got 50% as homozygote and 50% heterozygote, seen here, but they're all going to have blue eyes because this big B is dominant over this little b. So the phenotypic ratio is 4:0 and the genotypic ratio is 1:1. So moving on, we're going to apply this to a test cross. So we might have an organism which is dominant for a particular gene, but we want to see whether it's heterozygous or homozygous. So we need to apply that. So we use a test cross. So this, to make a test cross, the organism whose genotype is being tested is crossed with an organism that is homozygous recessive. So we're just going to do one of those. So a red-eyed fly and a homozygous recessive blue fly were put through this cross. So we were trying to determine whether that red-eyed fly is a heterozygous or a homozygous dominant. So big B question mark and little b, little b. So we're going to move these in. And then, same as last time, apply it. So out of the offspring, 50% had red eyes and 50% had blue eyes. So therefore, that red-eyed fly must have been heterozygous at the eye-coloured gene. There we go. So we had 50% heterozygous and 50% homozygous.